Welcome to Mr. Chalk's Revision Tips. In this video, we will look at
Transcriptional control, post-transcriptional control, and post-translational control. So, gene regulation is required for cells to specialize and work in a coordinated way. So, there are many ways in which genes are regulated, categorized by the level at which they operate. So we can have translational control, so genes can be turned on or off. We can have post-transcriptional control, so messenger RNA can be modified, which regulates the translation and the types of proteins produced. We can have translational control, so translational control can be stopped or started, and post-translational control, so proteins can be modified after synthesis, which changes their function. So most mechanisms that control gene expression do so by controlling transcription and synthesis of messenger RNA. Chromatin remodeling is a dynamic modification of chromatin architecture to allow the access of condensed genomic DNA to the regulatory transcription machinery proteins and thereby control gene expression. So, in molecular biology and genetics, transcriptional regulation is the means by which a cell regulates the conversion of DNA to RNA. So, it can do this in a number of different ways. So, you can have a transcription factor. So, this is a substance such as a protein that contributes to the cause of a specific biochemical reaction within a body process. A promoter, so this is a region of the DNA that indicates transcription or initiates transcription of a particular gene. You've got sigma factors, so these are specialized bacterial cofactors that complex with RNA polymerase to encode for a sequence specifically. You can have co-activators, so these are proteins that work with transcription factors to increase the rate of gene transcription. And you can have co-repressors, so these are proteins that work with transcription factors to decrease the rate of gene transcription. Heterochromatin is tightly packed or a tightly packed form of DNA or condensed DNA that comes in multiple varieties. So these varieties lie on a continuum between two extremes of being or allowing the genes to be fully available to the cell and fully available for transcription, or where the genes go and they wound up that tightly that no transcription can take place. So this tightly wound DNA causes chromosomes to be visible during cell division. The transcription of genes is not always possible when DNA is tightly wound because RNA polymerase can no longer gain access to these genes. Protein synthesis does not occur during cell division but during the interface between cell divisions. So this is a simple form of regulation that ensures that proteins necessary for cell division are synthesized in time. It also prevents the complex and energy-consuming process of protein synthesis from occurring when cells are actually dividing. So DNA coils around histones because they are positively charged and DNA is negatively charged. So histones can be modified to increase or decrease the degree of packing. So the addition of methyl groups makes the histones more hydrophobic, so they bind more tightly to each other, causing the DNA to coil more tightly and preventing the transcription of genes. Histone phosphorylation is a transient histone modification that becomes introduced by extracellular signals such as DNA damage or the entry into mitosis. And the addition of acetyl groups or phosphate groups reduces the positive charge on the histone, and this causes the DNA to code less tightly, allowing certain genes to be transcribed. Post-transcriptional control can occur at any stage after transcription, including RNA splicing, nuclear shuttling, and RNA stability. So once RNA is transcribed, it must be processed to create mature RNA that is ready to be translated. So the product of transcription is a precursor molecule, right, which we call pre-mRNA. So this is modified, forming mature messenger RNA that can bind to a ribosome and code for the synthesis of a required protein. A cap is added to the 5' end and the tail is added to the 3' end. These both help stabilize the messenger RNA and delay degradation in the cytoplasm. The cap also aids binding a messenger RNA to ribosomes. Right, splicing also occurs where RNA is cut at specific points, so the introns are removed and the extrons are joined together. Both processes occur within the nucleus. So, like we previously said, the product of transcription is a precursor molecule. This is modified, forming mature messenger RNA that can bind to a ribosome and code for the synthesis of a required protein. So, these have the same effect as point mutations and result in the synthesis of different proteins which may have different functions. So this increases the range of proteins that can be produced from a single messenger RNA molecule or gene. Translational regulation refers to the control of the level of protein synthesized from its messenger RNA. This regulation is vastly important to cellular response to stresses, growth clues, or differentiation. So there are a number of ways that this can happen. So we can have degradation of messenger RNA. So the more resilient the molecule is, the longer it will last. So the greater the amount of translation that will happen. We can have binding of inhibitory proteins to messenger RNA, which prevent it binding to ribosomes and therefore stop proteins from being synthesized. And we can have the activation of initiation factors, which aid to the binding of messenger RNA to ribosomes. Post-translational control can be defined as mechanisms by which the protein structure can be altered after translation. Proteins are polymers of amino acid, and there are 20 different amino groups or different amino acids. So both the order and identity of these amino acids are important for the role that the protein plays within the cell. So the addition of a non-protein group, such as carbohydrate chain, lipid, or phosphate, can affect this action. Modifying amino groups and the formation of bonds such as disulfide bridges can affect them. Folding or shortening of proteins will have an effect on that protein action. And the modification by cyclic AMP. So for example, in the lac operon, cyclic AMP binds to a cyclic AMP receptor protein, increasing the rate of transcription of structural genes.
Thanks for watching.